Assalamu alaikum. This is Muhammad Hassan Shafiq, and I welcome you all to the online biology program for grade 12. Today, hopefully, will be the last lecture of this important chapter, which is chromosome and DNA. And in today's topic, we will be talking about translation, which is the continuity of the topic gene expression, and we will also be talking about mutations, the types, the importance, and the diseases which can happen if mutations happen. So starting off today's lecture, we will be talking about translation first. Translation is the second step of gene expression, and in translation, messenger RNA produced by transcription is decoded by the ribosome to produce a specific amino acid chain or polypeptide that will later fold into an active protein. So the messenger RNA, which was formed in transcription, has now traveled to the cytoplasm, where it is decoded by the ribosome according to the instructions it carries to synthesize a specific polypeptide, which will later fold into an active protein. Although transcription is a continuous process, but for convenience, we will discuss it in four phases: the activation of amino acids, the formation of initiation complex, the elongation, and termination. So starting on with the first step, which is the activation of amino acids. The activation of amino acids refers or actually means that the binding of free amino acids dispersed in the cytoplasm to the 3' end of transfer RNA molecules. Like amino acids have to be brought to the specific site for protein synthesis to happen. They have to be activated in a way like they have to be brought by initially binding to the 3' end of a transfer RNA molecule, which in this way will form a complex called the amino acyl tRNA complex. This binding is catalyzed by amino acyl tRNA synthase, which is the activation enzyme. Various amino acids that are to take part in polypeptide formation have been continuously activated throughout the process of transcription. Now, if I very quickly go through this figure 23.16, which I'm just talking about, we have an amino acid which is acted upon by an enzyme called amino acyl tRNA synthase. This binding is catalyzed and then it leads to an amino acyl AMP complex after the removal of two phosphate groups. This forms a tRNA molecule with a free 3' hydroxyl end, which will be acted upon by the enzyme, and we can now see that the OH is now replaced by that specific amino acid molecule, right? So this is the activation where an amino acid gets converted uh, after binding to a tRNA to form a complex called the amino acyl tRNA complex with a 3' end. Instead of having a hydroxyl group now, a free hydroxyl group now, we have a specific amino acid which is according to the instructions in the messenger RNA. Next step is the next phase in translation is the formation of initiation complex. In the process of translation, uh, it actually begins with the formation of the initiation complex. It is formed by the combination of ribosomal subunits like a messenger RNA and first amino acyl tRNA complex. First, a tRNA molecule carrying a chemically modified methionine to the smaller ribosomal subunit. This binding is controlled by an enzyme called initiation factor. At the same time, 5' end of the messenger RNA molecule also binds to the smaller subunit of ribosome with the help of another initiation factor. Initiation complex is completed when larger subunit of ribosome is also placed upon smaller subunit. So after the initiation complex, what is happening is that the combination of ribosomal subunits The messenger RNA and the complex are like they chemically combine. The first tRNA molecule is usually a chemically modified methionine, which binds to the smaller ribosomal subunit controlled by the initiation factor. At the same time, the 5' end of the molecule also binds to the smaller subunit of ribosome with the help of another initiation factor. So we have two initiation factors here. Number one, the binding of the tRNA to the smaller ribosomal subunit, and the same time the binding of the messenger RNA molecule to the smaller subunit as well. Initiation complex is formed like you can see in figure 23.17 when larger ribosomal subunit is also placed upon the smaller ribosomal subunit. Like we do know that ribosomes have two subunits which in eukaryotes are 40S and 60S respectively. The region of smaller ribosomal subunit where first amino acyl tRNA complex is attached is called the P site, P for peptidyl. Here peptide bonds will be formed between the successive amino acids during elongation phase. Nearly two other sites are also established, the A site and the E site. The A site stands for the amino acyl site like you can see in figure 23.17. The amino acyl site is where the successive tRNAs will bind, and the E site is where the empty tRNAs, after giving their amino acid, will then be released from the ribosome during the elongation process. So let's just quickly talk about this again. The three sites are the P, A, and E site. The P is where the peptidyl bond is formed. Uh, the A site where successive tRNAs bearing amino acids are attached, and the E site where the amino acid will be exited from the ribosome. Now let's talk about the chain elongation or the polypeptide elongation here. In this phase, ribosomal units move along messenger RNA. Amino acids are brought by tRNAs which are joined later to form a polypeptide chain. So we can see that an amino acid comes at the A site and forms a bond between the amino acid it is carrying with the amino acid already present at the polypeptide chain on the P site. A bond is formed and the ribosomal unit moves ahead with the P site now becoming the E site, and the amino acid will then be released. Whichever codon of messenger RNA is exposed at the A site, its anticodon bearing amino acyl tRNA complex will bind to it with the help of an enzyme called the elongation factor. There's an enzyme called the peptidyl transferase which emerges from the P site, and this removes the amino acids from the tRNA present on P side and binds it to the new coming amino acid with the help of a peptide bond. The ribosomal subunits slightly move along messenger RNA from the 5' to the 3' direction so that a new codon is exposed at the A site. So moving ahead, the initially what was the A site is now the P site where the new peptide bond will be formed, and where there was initially the E site is now the P site. So this is how the ribosome keeps moving along the length of the messenger RNA. It moves from the 5' to the 3' end, and this movement is called translocation. As a result, an empty tRNA is reached at the E site after giving its amino acid and now is ready to leave, so that another amino acid can now come and bind at the exposed A site. The three steps are repeated again and again until the ribosome reaches the end of the messenger RNA sequence at its A site. Termination, the uh, termination is the last phase where elongation continues to move until a chain terminating stop codon is exposed at the A site. Stop codons do not bind to any tRNA, but they are recognized by release factors that terminate the process of translation, and the polypeptide is released from the tRNA. The tRNA is released from the ribosome, and the ribosomal subunits get separated from the messenger RNA. So if I can explain this termination process diagrammatically, you can see that the growing polypeptide chain reaches uh, the end where there is uh, a release factor. Stop codons do not generally bind to any tRNA, but they are recognized by release factors, and the polypeptide gets eventually released, like you can see in step 3. The transfer RNA is released from the ribosome, and the ribosomal subunits no longer remain attached to each other and are eventually separated. If I differentiate it in the form of a table between the translation process in prokaryotes and eukaryotes, you can see that the prokaryotes ribosomes are smaller while the eukaryotic ribosomes are larger. The prokaryotic translation occurs in the cytosol, while it can also occur in the rough endoplasmic reticulum as well in eukaryotes. The initiating amino acid in prokaryotes is N-formyl methionine, while in eukaryotes it's not modified, it's simply methionine. Only a single initiation factor is there in prokaryotes, whereas more than one is present in eukaryotes. Two release factors are used in prokaryotes, while a single release factor is there in eukaryotes as well. Moving on to the last topic of this chapter, which is mutations. Mutations, we have talked about this in earlier classes as well. I'm sure you pretty much know what mutation is. A mutation is a permanent change in the DNA sequence that makes up a new allele in the population. They range in size from a single DNA change to a larger segment of or a chromosome being sometimes changed, resulting in uh, an overall change in the number of chromosomes in a cell as well. These agents which cause mutations are called mutagens, while the organism or cell which undergoes mutation is called a mutant, which is now very different from its original wild type form. Sources and types of mutations. There are many sources of mutations which are basis different types of mutation. The sources of mutations may be physical, chemical, or biological. The physical mutations include radiations of any kind, for example, the UV, gamma, or X-rays, radioactive rays, high temperature, and high temperature alterations. These all actually are physical mutations. The chemical mutations include colchicine, mustard gas, nitrous gas, aquidine orange, radioactive isotopes, and free oxygen particles. Now, I request you all to do remember at least 3 examples for each mutagen type. The third source is biological, which may be induced by living organisms like or living particles like viruses, transposons, and errors that occur during meiosis or DNA replication. So all these 3 different sources can chemically or physically or biologically alter the genetic makeup, which will result in an overall different reading expression of the DNA, hence a mutation. Types of mutations can either be point or chromosomal mutations. The origin of mutation is called mutagenesis. Based on this, there are 2 types. Number 1, spontaneous mutations, which occur automatically due to internal or external factors. The other mutations are called the induced mutations, which are produced artificially for the establishment of new varieties of organisms. So these are done intentionally. The mutations are of 2 types depending upon where the mutation occurs and to what extent. A mutation that results in a change of a few nucleotides but does not overall affect the reading pattern is called point mutation. The mutation that cause change in the structure, the other form which causes a change in the structure of number of chromosomes, are called aberrations or chromosomal mutations. Importance, it may be useful or harmful. Harmful is that it might decrease the fitness or survival level of a species in the environment. Like there are many examples like born without part of a brain. A cancer is also a form of a mutation. The developmental abnormalities such as microcephaly, cleft palate, Cases of abnormal number of chromosomes like Down syndrome, Klinefelter syndrome, and Turner syndrome, which we will talk about later, and hereditary disorders like sickle cell anemia and phenylketonuria. So definitely the harmful mutations, the harmful aspects of mutations are definitely there. They can result in diseases. They can overall reduce the survival chances of a species in an environment, or they can also result in compromised individuals as well. The useful aspects are also there is that mutations are one of the biggest contributing factors towards evolutions, which also at one hand increases the uh, adaptability of an organism to its environment. Changes in the genes controlling development can have major effects on the morphology of the adult. Because these organisms or these effects are so significant, scientists suspect that changes in the developmental genes have helped bring about large-scale evolutionary transformations. Mutations are considered the driving force of evolution, where less favorable mutations are removed, and ultimately we are born with the mutations which were generally selected, more favorable, and have gradually helped us to be better adapted to the environment. Like throughout history, you can see that there were many epidemics in the past which wiped out quite a large population of humans, but now those diseases are, even if they are still prevalent in today's world, are not as dangerous as they used to be in the past, maybe due to us being more adapted or becoming more as resistant due to evolution. And in that case, mutation has played a very important part as well. Talking about chromosomal mutations or aberrations, they are the changes in the number of chromosomes. Here we will discuss symptoms, causes, and possible treatments of some of the chromosomal mutations. Down syndrome, Klinefelter syndrome, and Turner syndrome are very important, and since these are in the book, we will be talking about these. Down syndrome is characterized by an extra chromosome. So we have an extra copy of the 21st chromosome. Symptoms include, you can see trisomy 21 in the diagram figure 23.23. So the symptoms include abnormal shape, small sized head, excessive skin at the back of the neck, a flattened nose, single crease in the palm of the hand, white spots on the colored part. So there are definitely physically abnormalities seen and quite evident. In this case, the 21st chromosome fails to segregate, resulting gamete with a 24th chromosome. The fertilization of such an egg will definite with a 23, with a sperm having 23 will result in a child having 47 chromosomes, which is 2n plus 1. There are currently no treatments for this syndrome, but it can be managed to some extent by measures like regular checkups, medication, surgery, counseling, and support. Klinefelter syndrome is also characterized by 2n plus 1, but in this case, you can see the case is 44 autosomes plus an XXY. The symptoms include, since we have two X chromosomes now, it includes feminine characteristics, although they are males. They have wide hips, the testicles remain small, they have sparse body hair and an enlarged breast. It is caused by non-disjunction of a sex chromosome pair during maternal meiosis. The treatment can be like they can be given testosterone for sexual development in males around the age of puberty. It can have help a boy have more normal development. The fertility is still quite compromised, and the treatment usually continues throughout a man's life. You can see in figure 23.24 that it is actually having an extra X chromosome, so there is non-disjunctions during maternal meiosis. So in this case, it will also result in infertility. The third chromosomal aberration mentioned in the book is the Turner syndrome, which is a case of 45 or 2n minus 1, which results in 44 autosomes plus an X naught. Naught represents a null gamete having no chromosome. So in this case, the symptoms include a shortened height, abnormal bone development, a larger than usual number of moles, edema or extra fluid in the hands and feet, causing puffiness, and it is caused by non-disjunction of sex chromosome pair during maternal meiosis. So it is caused by non-disjunction of a sex chromosome. As you can see in figure 23.25, treatment is growth hormone can be given to improve height and growth, and it can influence a girl's final adult height, especially if treatment is started. Estrogen replacement therapy helps the girls to develop the physical changes of puberty, including breast development and menstrual periods. The treatment is often started when the girl reaches about age 12 or 13. So, point mutations are also there. Just to mention a few, we can talk about sickle cell anemia. You've heard about this disease in previous classes as well, where the blood is not able to bind sufficient oxygen, and a case of anemia or paleness prevails the body. Blood transfusions can be done. It is caused by a single mutation in where the glutamic acid is replaced by valine, mentioned on page 205. So oxygen binding capacity is severely reduced. Bone marrow stem cell transplant still appears the best cure for sickle cell anemia, and folic acid supplements might also be given. The second disease mentioned in the book regarding mutations is the phenylketonuria, which I mentioned, and it is caused by the inability of the body to break down the amino acid phenylalanine, called phenylketonuria. The symptoms include light skin, hair, and eyes, mental retardation, seizures, and skin rashes. Phenylalanine can be converted into tyrosine by the enzyme phenylalanine hydroxylase. And due to a point mutation, the enzyme becomes defective, and phenylalanine converts phenylalanine into toxic phenylketones, which get accumulated in the nervous system, resulting in mental retardation. And this is caused by an autosomal recessive gene, and one can get the disease if it is inherited from both the patients. Treatment includes a diet that is extremely low in this amino acid phenylalanine, especially when the child is growing. Lopinavac is made from infants, is made for infants with the phenylketonuria. It can also be used throughout life as a protein source that is extremely low in phenylalanine. So this ends today's chapter, and this was a very important chapter. I hope that you had a good understanding. You are all requested to give this chapter a thorough revision, and this is very important from the examination point of view, as a lot of questions are usually asked in a federal board from this chapter. This was all, and I'll continue with you in the next lecture.